I'll start by saying I started my PhD in 2015, so I've been working in cancer research, mainly lung cancer research, and specifically the project we'll be talking about today for about the last 10 years. And about 5 years ago, in 2020, I had the great luck to come across Yun-Hsin, who was then a master's student. We've been working together for the last 4-ish years, and she, is deservedly taking up a PhD spot in October. So yeah, as Lynn mentioned, you get 2 for 1 today. So I'll present the first kind of half, which is kind of more the development of this new biomarker we have. And Yun-Hsin will take over for the second half.
So I'm a bioinformatics research assistant in UCL. So I've been working with Dhruva since, I think, 2020. Yeah, it was a long time. I've been focusing on lung cancer, and I followed the project to look at this novel biomarker that we can hopefully implement in lung tumors. So yeah, it's very exciting work. And yeah, so I'll hand over to Dhruva to start the presentation.
So with me, who's dual trained and have a medical degree, I think it's always important that we're kind of doing scientific work, but to really keep the patient at the heart of the story. So let's start with the patient story. And a separate thing I would say as well is, we'll talk about this. We're really grateful for the opportunity to present. I think it's important for us not just to do science, but also we have a responsibility to communicate back, not just to the scientific community, but to patients. And so we'll do that. But also it's kind of deeply personal work for both of us. So my grandfather passed away from lung cancer during my first year of medical school. So it's not arbitrary at all that I work in this area, and yeah, it's very meaningful for me. So here's a patient with lung cancer that will kind of follow their story during this slideshow. We call them John, and as a newly diagnosed cancer patient, this is a question that John might have. What are my chances of seeing my grandson's 5th birthday? And maybe it's a question many patients have when they're newly diagnosed with lung cancer. They want to know how their life is going to change, what sort of outcomes they're going to have. And this is, as everyone here knows, is a kind of growing problem and present around the world. Oncologists also have a similar sort of question, and their question is more framed around treatment options. So I know often from working with different patient groups that patients can often know more about their cancer than certainly their primary doctor and probably even their oncologist at a certain point. So this isn't aimed to be patronizing at all. And we have a focus here on non-small cell lung cancer and the lung adenocarcinoma subtype. But to kind of zoom out and just keep it simple. So yeah, the oncologist might ask, will John achieve survival benefit from chemotherapy? And the way that's currently looked at the moment is John will go for a CT scan. And on the CT scan, the size of his primary tumor will be measured with a ruler, and also the extent of the spread. So the size of the primary tumor is T, N is spread to nodes, N is distant metastases. And based on that, there's an assessment of, is this person high or low risk? John is considered low risk, then surgery alone may be a great treatment option where the cancer is removed and there's no need for any other radiation therapy or drug chemotherapy. Or if from this CT scan and TNM staging, if John is perceived to be high risk, then the oncologist might think that John might benefit from surgery to remove the primary tumor and also chemotherapy. And this is obviously a joint discussion based on what the oncologist sees and John's preference. So there's a lot of uncertainty in the field about how we should manage stage 1 lung cancer. And we'll go on to that in the next slide. But here I just want to highlight that this is a problem that's not going away. So the proportion of stage 1 diagnoses is increasing. So at the moment, it's about 15% of all lung cancer diagnoses. But if we move to a screening strategy, so at the moment, in the UK, how lung cancer is diagnosed is someone like John might be symptomatic, have weight loss, be coughing blood, go to see their primary care doctor. And in that sort of group, cancers tend to be diagnosed in stage 2, stage 3. Whereas now in Germany, maybe now in the UK, and certainly in America, they're starting to think about doing CT screening. So this is people who are considered higher risk, maybe with some level of family history or smoking exposure, to kind of proactively invite people in for a CT scan, which, the trials have shown that's a very good strategy. And actually, firstly, you pick up cancers earlier. And because the cancers are picked up earlier, they're more treatable. However, for this specific problem of kind of diagnostic uncertainty in stage 1, that problem is growing with the screening strategy. So this is the current pathway again. And with this pathway, so here we have a survival curve. So, again, I want to say this is kind of like meaningful and personal research for us, and sometimes presenting stats like this can seem very impersonal. This is just kind of aggregate data and helps us get an idea of what the problem is and where we can move the needle. So at the moment, a patient like John, 5 years after diagnosis, if diagnosed with stage 1, 25% of patients sadly succumb to their disease. So this strategy of surgery alone may not be the best option. So the idea would be to develop another test to integrate with staging, maybe based on the genomic profile of the tumor. And then you can more accurately classify what the true risk is. And this is what that pathway might look like. So at the point of diagnosis, there's a CT scan, a biopsy tumor of tissue is taken, and that's gold standard everywhere in the world. So that biopsy is required to look at the tissue down a microscope and see, are these cancer cells or are they normal cells? And then do tests on top of that. So one test might be staining to look at, is it adenocarcinoma, squamous cell carcinoma? There might be mutation tests to look at the EGFR mutants, KRAS mutant, etc. And also there's this emerging idea of doing a genomic RNA test, which specifically predicts risk. So a validated RNA test could reduce undertreatment of stage 1 lung cancer by going beyond just saying in the gray line, stage 1, to on the right, the blue line, which is low risk stage 1, maybe surgery is the best option. Whereas high risk stage 1, just because the tumor looks small on the CT scan, doesn't mean this person couldn't benefit. So this has been a pretty well-defined problem for at least 25 years. So in the early 2000s, with the advent of kind of more genomic analyses, so genomic sequencing, things like that were becoming cheaper and more accessible, and people are starting to recognize that cancer is a genetic disease. Lots of people tried to solve this problem. So, from 2000 to 2010, there were at least 50 publications in this area. But so far, in sitting here in 2025, there's not a single test that's approved, FDA approved, or approved by the NHS in the UK. So why is that? So one reason is poor reproducibility. So here what we're showing is this green cohort is the cohort used to develop the test, and the test works in that cohort. And this blue cohort is a separate group of patients, and the test doesn't work in the cohort. And so when you see something like this, that there's a failure for the test to validate in external groups of patients, what that means is that it might be highly tuned to the training population. Maybe it's tuned to things like diet or ethnicity, and it's not trained to something robust, some kind of features of the tumor aggressiveness, which is what we would actually want it to be tuned to. And the second reason is limited utility. So as we discuss, the kind of question mark is really over stage 1 and the use of chemotherapy in stage 1. And so a test in stage 2 or stage 3 cancer, which says this person is high risk and would benefit from adjuvant chemo or radiotherapy, that's actually not useful beyond the existing standard of TNM staging. And all the tests that have been developed to date fail to validate in patients with stage 1 lung cancer. So it's really, when I was starting my PhD in 2015, this was kind of the playing field. Lots of people have tried to solve this problem, diagnostic uncertainty, stage 1 lung cancer, and none of them have succeeded because their approaches are not reproducible and because they don't add utility to the existing clinical standard. So we approach this from a fresh evolutionary perspective. So this is winding the clock all the way back to Charles Darwin in the Beagle, when he was a young scientist and kind of traveling the world and trying to figure out what he wants to do in his life. And as you probably all know the story, that he was floating around the Galapagos Islands and noticing that each of the islands would have species of finches, but the finches would have a different beak shape. And that would be linked to the type of food available to the bird. So if the nut, for example, was more deeply buried, then it might have like a longer, thinner beak. And if the nuts were kind of harder, it might have a larger beak, able to crack the nuts. So he kind of came up with this idea that actually species aren't fixed, and it's not that from time immemorial that all the species have been designed exactly as they are. Actually, there was a kind of common starting point, and with different selection pressures, these finches, which came from a common ancestor, developed different beak shapes adapted to their environment. So that was in 1837. In the 1970s, Peter Noel was looking at this stuff and saying, maybe what we're seeing operate at the level of species and individual organism, maybe that also operates within the level of an individual cancer between different groups of cancer cells. So he wrote, acquired genetic lability permits stepwise selection of variant clones and underlies tumor progression. So what he really means by that is that maybe to begin with, there's a single genetic insult to the cancer cell that allows it to divide quicker. Yeah, let's have a look at this cartoon figure. So at the beginning, it's a normal healthy population, and then we get this blue subpopulation growing out. So these cells, whether it's 10 cancer cells, 100, or 1,000, they all have exactly the same genetic properties. And now we can see this brown subpopulation branching off a bit like in Darwin's diagram. So this brown subpopulation might have slightly different properties. And as the axis of time winds on, we can see other subpopulations emerging, which might have different properties. So this purple subpopulation has the capacity to metastasize and spread around the body, which the brown and blue clones didn't have that ability. On the x-axis, we can see the point of diagnosis. So at the time a cancer is diagnosed, it typically consists of 1 billion cancer cells. And up until the 1970s, the prevailing belief is that all 1 billion cancer cells would be exactly identical. Whereas now we know, and has been seen time and again in many different cancer types, that's not true at all. Depending on where you sample the tumor. So here in this cartoon at the bottom, if we take a cross section at one point in time through the tumor, we can see that it's not one homogeneous population, it's actually lots of different communities, which all have slightly different properties. So some might be better at invading, some might be better at evading the immune system, some might be better at kind of introducing vasculature, growing blood vessels to deal with the shortage of oxygen. Some might be better at spreading around the body. So this is the kind of problem that we need to face. And Gloria Hepner wrote in 1984 this concept of intratumor heterogeneity, that within a tumor, we have these different cell populations. Tumor cell differences are believed to be due to the presence of distinctly different subpopulations capable of reading true. In our own context, when we think about biomarkers and why these tests aren't working, why they're not reproducible, and why they're not adding medical utility, this may be the reason that when we take a diagnostic biopsy of a tumor, we typically sample less than 1% billion cell community. And if all their cell communities are different, maybe we're going to get a different readout depending on where we place the biopsy needle. This slide speaks to the genetic basis of what's happening here. So to begin with, every cell in our body, except for the sex cells, are diploid. So we have 23 chromosomes, and we get one copy of each chromosome from our mum and one from our dad. We have 2 copies of each chromosome. But as a cancer evolves, we get genetic abnormalities. So here in this blue subpopulation, you can see we've gained an arm of the chromosome 1. And in this brown subpopulation, we've lost a copy of chromosome 2. In the purple one, on the contrary, we've gained a copy of chromosome 2. And in this red one, we have 4 copies of chromosome 2. And so this speaks to why the cancer, depending on which clone and which portion you sample, will have different properties, and because there's a different genetic architecture. So some of these changes will be present clonally, i.e. in every cancer cell, you get the same picture. So that change that happened for chromosome 1 happened very early in the cancer's history, and that would be clonal. Whereas other changes occur subclonally. They occur later if you kind of think about the evolutionary tree. So some are clonal in the trunk of the tree, that they're present everywhere. Others are subclonal, that they're in the branches, so they're present in some parts of the tumor, but not. And our lab in 2017 published in the New England Journal of Medicine a study where they profiled 100 lung cancers. They looked deeply at the genetic changes, the DNA changes, and they found that 50% of changes were clonal, i.e. were shared through all the cells of each person's tumor, and 50% were subclonal, which has big implications for treatment choices and for our projects developing a better biomarker. Taking this idea of evolution, taking this idea of biomarker, what's the interface and where do they meet? So this kind of leads on to my own work, which we published in 2019. So as we all learned in school, from DNA comes RNA. And it's this RNA which is actually the substrate for genomic tests. So when the pathologist is in the lab, they have a sample of John's tumor biopsy, and they're trying to get a readout. Is this an aggressive tumor or not? At the molecular level, they'll be siphoning off some RNA and testing the RNA. And they'll be using that to consider mortality risk and then go back to the oncologist so John and John's oncologist can make a combined treatment decision. So here in the cartoon, if we're looking at depending on which part of the tumor we biopsy, we can get a completely different result if we use these subclonal changes, which are shown in the green. So in the top right, we can see we have 4 copies of that gene. So if the tumor biopsy needle was placed in that part of the tumor, we would think this is a high-risk tumor. Whereas in the brown part of the tumor, if we were to place our biopsy needle there, there's only 1 copy of the green gene. So we consider it's low risk. And this is this concept of tumor sampling bias. Sampling 2 regions of the same tumor from the same patient can give 2 completely different results. So in my work in 2019, we looked at this in a cohort of patients with lung cancer, and we found that using existing tests, all these tests that have been published from 2000 to 2010, in half of the cases, using the published test, we get a different result depending on where we place our biopsy needle. And this may well be why they're not working, because it's kind of, you're at the casino every time you use the test, depending on where the needle was placed. So that was kind of interesting, but that's just science. That doesn't actually help anybody. So we wanted to go further and we wanted to see, well, we've identified that this is a problem, that depending on where you place your biopsy needle, you get a different result using existing tests. Can we solve that problem? So using insights from Darwin, there's nothing new in science. Everything has kind of already been discovered. We reasoned that, well, rather than kind of focusing on these changes that are subclonal in the branches of the tree, why don't we focus on changes that are present in the trunk, that are present in every part of the tumor? So in this case, if we focused on the clonal alterations, we can see there's the same number of copies of DNA for the chromosome. There's the same number of copies of RNA, 3 versions of the transcript. And so when it comes time to calculate our genomic risk, we get high risk in every part of the tumor, which would be the correct result for this patient. So this was our hypothesis, that if we focus on clonal or homogeneous or early evolution genes that are expressed uniformly throughout the tumor bulk, could this solve the sampling bias problem and yield a better biomarker? It's not subject to the problems that kind of torpedoed the previous approaches. So here's the idea again with this attractive cartoon that Fosfo put together for us. So here at the bottom, this very colorful ball, it's a tumor. The idea is that's 1 billion cells. That's when the tumor is typically diagnosed. And you can see it's all different populations of cells, some purple, some blue, some green, some yellow, etc. And that speaks to intratumor DNA heterogeneity, that speaks to unstable chromosomes, and which gives rise to kind of an unstable level of RNA expression. That at some points in the tumor, we get a very kind of high, like mountain expression. At other places, we get a trough, and it will completely bias and throw off our diagnostic results. So our hypothesis was, well, what about if we use Darwin's insight to focus on genes that are homogeneously expressed? So regardless of where we biopsy the tumor, rather than just looking at the pool of all human genes, which are 20,000 expressed genes, why don't we just focus on genes that are uniformly expressed everywhere in the tumor? In our analysis, we found that represented about 1,000 of those 20,000 genes. And from those 1,000 genes, we focused on genes that were robustly associated with survival, i.e. that they're clonal and they're kind of prognostic, to develop a test which we call Oracle. And using Oracle for this specific challenge of tumor sampling bias, we found that the proportion of lung tumors subject to sampling bias went from 50% using previous approaches to 11%. But that's kind of all well and good, but a critical person might say, well, you've defined this problem of sampling bias and you show that you've solved it, but you haven't done anything for the patient. You haven't actually solved the 2 big challenges. So let's look at them. And so the first question was reproducibility. And so we developed Oracle in this data set, which is very famous, rightly so, the Cancer Genome Atlas, which is all developed in the USA. So that features 470 patients with non-small cell lung adenocarcinoma. So here, all the survival information was developed in that USA-based cohort, and we tested it in this separate cohort from our collaborators in Sweden. So previous approaches, they wouldn't withstand this test. They would fail. We don't know why. We assumed the differences between the Swedish population and the American population, because they're not actually trained on robust kind of cancer aggressiveness features. Whereas we found Oracle does associate with survival across cohorts, and having a high Oracle risk score was associated with a threefold increased mortality risk. And this was independent of known factors. So our collaborators at Sweden, they really went through their record books for their cohort of patients, and they provided lots of information. So clinical stage, smoking history, World Health Organization performance status, age, sex. These are all things that are known to either strongly associate with survival, like stage, or weakly associated, like age or sex, I thought to weakly associate with survival benefit. And the idea is these tests are obviously expensive, and they also require some amount of extra tissue. They require some amount of pathologist time. But to justify all that kind of expenditure, there needs to be kind of a performance boost compared to existing risk factors. Oracle was shown to do that. And we went beyond the Swedish cohort. So we accessed a bunch of published data sets. And here I'm trying to differentiate from the work we did in 2019 from the work that Yun-Hsin will present shortly. So here we're looking at, this is retrospective. So this is data that people had developed at any time prior to 2019. And we downloaded the data with permissions, ethical approval, etc., from more than 900 patients in total, from 5 cohorts from all around the world, some from Japan, some from Europe. And we found that Oracle risk score was significantly associated with survival in all these different groups. But, as I keep bashing on, does this help John? So he's the person that we started with. So this is the current framework, as we say, the CT scan, the tumor staging, mortality risk. And based on that framework, stage 1 would be surgery alone. Yep. So here with substaging, this is the current best that we have in terms of pathology. So the difference between stage 1A and 1B is based on the tumor size. I believe it's something like 2.5 centimeters and below is 1A, and 2.5 to 3.5. I may be slightly wrong on those numbers, and they've changed since we did this work in 2019. But those are the sort of margins that would give you a classification of 1A versus 1B. And you can see in this small group of patients, substaging is just not informative for survival. 2 curves cross. So if someone is 1A, you don't know if they're going to do better beyond. Whereas in Oracle, the low-risk patients had better survival outcomes than high-risk patients, which kind of points to that it is useful and it's adding value in the stage 1 subgroup. So maybe kind of getting closer towards that vision of we have the staging information and also the genomic risk that allows us to predict mortality risk and help John and his oncologists make more accurate treatment decisions. And yeah, lastly, when will this help John? So in 2019, we published this work. We were on the front cover of the journal in Nature Medicine. And we had some press. So the Times covered us. They had a front page piece. And that's all kind of nice for us, but it doesn't really help the patient community. So how do we actually move that to the patient community? So to go from a biomarker that's been developed in a research study to something that's clinically approved and your oncologist can order and request, there's a few steps. So the first step after the research study is to develop it as a clinical assay. And so, not to get into the weeds here, but there are a few differences in terms of research grade technology versus clinical grade technology, the kind of samples you might have frozen environment in a research grade study versus in a clinical grade study where there'll be kind of fixed formalin embedded in paraffin. And so we did that work in 2019, and we also had a patent approved, which is important for the kind of commercialization and the adoption of these tests widely for implementation. The step after that is clinical validation. So shortly I'll be passing over to Yun-Hsin, who'll be talking about this work that we've been doing since 2019. So there's 2 arms to this. A retrospective is looking at clinical grade samples in a very large group of patients, 1,000 across 3 European sites, and prospective, which Yun-Hsin will talk about. And if those 3 steps, the research study, which we've done, clinical assay development, which we've done, clinical validation, which Yun-Hsin will tell you about, if those 3 things are done well, and the test bears out, which ultimately we have no control over, we can just do the science to the best of our ability, and then nature will do the rest. If those 3 things work out and the test looks promising and like we could actually make a difference in clinical use, that's when we submit for clinical approval, and organizations like FDA can. So with that, I'll hand over to Yun-Hsin.
So thanks for Dhruva's introduction on the background of Oracle. And building on this discovery back in 2019, in the next section, I'm going to show the validation work that we've recently done to demonstrate that Oracle is a promising genomic test, and our findings on how it could improve the management of lung cancer. So it starts with the first question that we ask is how robust Oracle is. So Oracle was designed by a clonal approach that would pick out the genes that were clonally expressed. So that means that that gene was seen in every cell. And we wanted to validate that how many tumors will be affected by sampling bias if Oracle is used. We expect that Oracle can improve a sampling bias problem because it can it is consistent across the tumor. So to test this, we've got a lung cancer patient cohort that is funded by Cancer Research UK. So that's the cohort based in UK, where data of 158 lung tumors were available. And in this cohort, multiple samples were taken from different parts of this single tumor. So to know if a biomarker will be affected by sampling bias, we simply apply that biomarker to the samples of these tumors, and then compare the risk prediction of multiple samples. So a biomarker is less subject to sampling bias if the test results obtained from every part of the tumor is the same. So as shown here. So we've got high risk prediction across every part of the tumor. Well, if the tumor is more prone to sampling bias, we will get the test result that is inconsistent across the part of the tumors. So we then examine the consistency of Oracle risk within each tumor. And we also apply this method to other 6 published biomarkers to benchmark against the performance of Oracle. So when testing with Oracle, it turns out that 81% of the lung tumors have consistent risk results, as shown in the top here. We have consistent risk prediction across every part of the tumors, which ranks the top among all the 7 biomarkers that will be included in this analysis. So we validated that Oracle is a robust biomarker for lung tumors. So you might think next, does the robustness maintain in the association of Oracle with survival risk? So being the most important part of this study, we validate the survival association of Oracle in this independent cohort of 158 lung cancer patients who were recruited prospectively. And remember that all previous approaches for biomarkers, for biomarker design, have failed to be validated in external cohorts. So here we've successfully shown that Oracle is significantly associated with survival risk, and with the outcome means that patients with high Oracle scores have more than double the risk of death after 5 years from diagnosis. And this risk prediction power similarly remains significant even after accounting for the risk factors. We've shown similar analysis back in 2019. So here we again show the reproducibility of Oracle that is independent of these risk factors, such as the patient's age, sex, and smoking history, or the size of the stage of the tumor that were quite related to survival risk. We then also test Oracle in a subgroup of patients that are with stage 1 lung cancer. And in this subgroup of patients, they normally receive surgery alone, or even chemotherapy. The use of chemotherapy was quite controversial. So we found that the current staging method failed to distinguish the prognosis. So you can see that if we further classify the stage 1 patients with their tumor size to 1A and 1B, we didn't see much difference between their survival risk. However, Oracle can picks out a group of high-risk patients who might have improved survival outcome if we give the chemotherapy to this group of patients. So this result suggests that Oracle could potentially help the doctor make treatment decisions at the earliest stage of this disease. So here we have shown that how robust and reproducible Oracle is, but we want to gain more confidence by testing Oracle in a single biopsy setting, as this represents the routine clinical practice. So in routine clinic, we only take single biopsy from each tumor. So here we simulate a single biopsy cohort of 158 lung tumors by randomly taking 1 sample from each tumor. And with these samples, we then calculate the Oracle risk and then test the association with survival outcomes with these samples. So by repeating this sampling process for 1,000 times, so we've increased the numbers of testing to gain more confidence. We obtained a distribution of the test results of 1,000 test results. And the hazard ratio here we measure is measuring the risk of dying if Oracle score is high. So if we've got this hazard ratio larger than 1, this means that the risk of dying is increased when we have a higher Oracle risk score. So as we can see that in this analysis, Oracle remains informative for survival risk, even single biopsy is applied as in the real clinical practice. So far we've gone through what is Oracle and how Oracle predicts the survival. And we already know that Oracle is derived from primary tumors and high-risk signal is associated with the risk of survival. But is this high-risk signal detectable before tumor presents? Or is it only detectable only in the tumor itself? So to address this answer, we traced the origin of high-risk signal by applying Oracle to different development stages in cancer. So normally a cancer will start with the normal cells, and these cells, some of them might progress and gain a mutation to become precancerous lesions, and some of them might later become tumors. So we know that Oracle is already present we can already detect the Oracle risk in the tumor. But how about the others? So we found that this high-risk signal is also increased in precancerous lesions compared to the normal lungs. So this observation can be understood if we look at the genomic alteration during the cancer evolution that Dhruva has been showing a lot in previous slides. So at the normal stage, cells contain pairs of chromosomes. So that's the one from your mother and the one from your father. So as the cancer evolves, the cells will undergo chromosome chaos, where the numbers of chromosomes will increase or duplicate during the formation of tumor. And Oracle is picking up the clonal event, which is shown as the blue chromosomes here, that are already altered at the earlier stage during this evolution when the lesions are already formed. So this means that Oracle can stratify the high-risk lesion to provide an early intervention even before the diagnosis of the primary tumor. And how about predicting the events in years later after diagnosis? So the most clinical concern question might be whether the cancer will return, as they often come back with more aggressiveness. So we compare the rate of disease progression determined by Oracle risk groups. So we stratify tumors by the Oracle, and we found that in low Oracle tumors or low risk tumors, only 29% of them have disease progression after 5 years from diagnosis, whereas the rate surges to 89% in those detected with high risk in any part of the tumor. So this means that higher Oracle risk predicts a greater chance of cancer coming back. And also at the same time, we found that Oracle can inform the risk of cancer spreading across the body, which is metastasis. So how is this achieved? So from the evolutionary pattern, we can see that metastasis grows out from this purple clone that was found in the primary tumor. So we think of testing Oracle in this purple clone that's shared with the primary tumor, but also compared to these brown clones or other clones that did not grow into the metastasis. And we found that Oracle score in this purple clones that sees the metastasis is associated with higher Oracle score. So Oracle score is higher in this metastatic clone compared to the other clones that did not share with the primary tumor. And this means that Oracle can inform the metastatic potential. So together with previous slides, these findings suggest that if further validated, Oracle can not only help doctor predict the survival risk, but can also extend beyond this to predict the chance of disease progression before tumor invasion. And this is important to achieve early cancer detection, also intervention. So what else can Oracle do? So chemotherapy has been the mainstay for cancer treatment for many years, but it is still a challenge to predict the patient's benefit to adjuvant chemotherapy, especially in early stage lung cancer. So we wonder if Oracle is useful to predict the chemotherapy response. By comparing the Oracle score, by comparing the Oracle risk in the patients that did not receive chemotherapy to those received the treatment, we indeed found that Oracle captures the survival outcomes affected by chemotherapy. So as you can see in this probability of the survival outcome, we see that the high risk group of patients has similar survival compared to the low risk in the treatment group. And we also found this evidence using the anti-cancer drug sensitivity database. So we simply rank the lung cancer cell lines in this database by the Oracle scores, and then we link that to the sensitivity of drug and of different anti-cancer drugs. And in this graph, we've plot the relationship of drug response and Oracle. And if we focus only on the approved drugs for lung cancer circled in the black, we found that higher Oracle risk is associated with better response to chemotherapy, such as cisplatin, which is commonly used in the treatment of lung cancer. So why is this happening? To help us understand the mechanism, let's look at the cells. So inside normal cells, the chromosomes are in pairs, as mentioned. So each time the cell divides, it should maintain the correct number of chromosomes, which is two in each pair. However, cancer cell division is an erroneous process where cells can result in alteration in DNA, as shown in the cells at the top. So you've got this red labeled, means that there's some mutation in the chromosomes in this DNA, or you can have changes in the number of chromosomes as shown in the cells at the top, where the numbers of chromosomes has been duplicated. So if we link this to Oracle, we find that cells with unstable chromosome numbers have higher Oracle risk, whereas those with stable chromosome number tends to have low Oracle risk. So we conclude that Oracle is associated and has a positive correlation with the unstable state of chromosome, which we call it chromosomal instability. And this variation in the chromosome composition can shape the fitness of the tumor cells during evolution, just like what we have learned from the Darwin evolution theory. So if the chromosome status is too stable, that might be insufficient benefit for the evolution. While if the chromosome status is unstable to the extent that advantageous population is difficult to be preserved, and this can be lethal to the cell viability. So why is this related to the prediction of chemotherapy response? If we think of the mechanism of chemotherapy, it works by inducing errors to cell division, which leads to the extreme unstable chromosome state. And this is captured by high Oracle risk. So this could possibly explain how Oracle high risk can predict better response in patients who receive chemotherapy, as the cells in these patients have acquired unstable chromosome states that is triggered by the therapy itself. Yeah, so coming back to the timeline of when Oracle will help the patients with lung cancer. Now, the prospective validation has been completed. We've shown that Oracle can predict the survival of lung cancer patients and potentially help doctor make a treatment decision, especially in the stage 1 patients at the earliest diagnosis. And in the meanwhile, we are now preparing the validation of Oracle in a larger cohort with 1,000 patients that were collected across the Europe. So our next step after this is to compare the patients with Oracle high risk receiving standard clinical care. For example, the management of lung cancer that will apply to those lower stage of the tumor, and compared to those who were given with chemotherapy or more frequent monitoring. to see whether Oracle can help improve the survival. So hopefully this validation can lead us to a clinical approval of Oracle as a genomic test to be used in the future clinic. Here is the quote that Dhruva would like to share.
Yeah, thanks so much for your sentence. So we'll just finish with this. So this is a quote from my colleague and friend in med school, Chua Zhang, who has a deep lived experience of cancer. So all our work is around biopsies and trying to kind of maximize the information we get from them. So this quote, I think, is quite nice to finish on. Biopsies aren't simply pieces of tissue under a microscope. Although mere milligrams, they weigh much more heavily in the hearts of patients and their families. In a small way, they encapsulate hopes and dreams, vitality and sickness, life and death.
Yeah, so yeah, I think this is the end of our presentation. And yeah, we'd like to express our thanks to the patients, as we couldn't finish this work without they providing the samples to our research, and also the generous fund from Cancer Research UK to support our work, and also many supports from many labs from different institutions. So yeah, thank you for all your time to join this talk.
Thank you. Thank you so much. This is just